Okay, can you guys all see my screen?
Yep.
Great. Well, welcome to office hours for biochem genetics. Um, today's or this week's office hours are going to be focusing on lectures 6 through 11. And these are the four TAs who worked on it. Yuri, Ivan, Alex, and myself, Kostov or KP. Um, I wanted to start this off by basically handing out some helpful tips that helped me a lot when I was taking biochem last year, biochem and genetics. Um, Always focus on the colored text, the ones that are red, blue, green. They usually have the most important and high yield information. Obviously, read everything, but if you're in a pinch or you're trying to focus on very high yield stuff, always look at the colored text. Pictures are always very helpful, and they can help visualize pathways and different biochemical processes. Um, if you're trying to read through something and you're not really able to visualize or understand like how um, some kind of signaling works or anything like that, then there's usually a picture in the lecture slides to help you get a better understanding. And so pay attention to those pictures, and it can help better your understanding of that topic. And then the third thing is the metabolic map. Um, I'm not sure if you guys have gotten a chance to look at that yet, or if you guys have seen it in class. But that thing is really helpful for seeing all the different pathways for carbohydrate synthesis and breakdown proteins, purines, pyrimidines, all that you guys will get to eventually. And it's very useful for visualizing different pathways and how things work out. So definitely make use of that. It'll help you a lot on your exams. And I think you can use it on your exams as well. We did that last year, and I think it'll be the same for you guys this year. So with that, we'll be starting off with the first lecture, which I think is Yuri.
Yeah, so I worked on lecture 6 and 7. I'm gonna start off with lecture 6, which is overview of metabolic pathway. These are the objectives. You can take a look at it later. But basically, I'm gonna go over like a high yield stuff. Next. KP, can you go to the next slide? Okay, so definitely know the definition of catabolism and anabolism. This diagram is basically showing that so all these substances with the red arrow, they're broken down into one substance. In this case, is um, acetate. And those blue arrows are showing that from acetate, all these substances are made. So the red is catabolism, the blue is anabolism. Next slide. So the definition of catabolism is breaking down process, which is also conversion. And during this process, they release ATP. And when there's like too much ATP around, the catabolism is suppressed, meaning it's stimulated by low energy charge but suppressed by high energy charge. And anabolism is the basically like opposite of it. It's the synthesis process, divergent, and it requires ATP and stimulated by high energy charge. These are the class of enzymes. Um, don't worry about like examples. Just know the enzyme and what it does. Basically, I think these enzymes are like fair game. They could ask any of it on the exam, so make sure to know what they do. These are pretty self-explanatory, so if you guys don't have any question, I'll move on to next slide. Okay, and then you are required to know the difference between rate limiting, and committed steps. So a lot of people think these are the same, but it's actually not. They like to test the difference between these two, so make sure you know. Committed step is like it happens early in linear pathway. It's irreversible and highly regulated. While rate limiting is irreversible and it's the slowest reaction, requires high activation energy, and usually it's under very intense regulation. So all committed steps are rate limiting step, but it's not vice versa. Next. So let's talk about enzyme regulation. So long-term regulation, it changes the actual quantity of enzyme present in the cell, and this happens mainly through gene expression and protein degradation. Induction increases enzyme synthesis, repression decreases it. And enzyme degradation happens mainly in two ways: the ubiquitin proteasome pathway for damaged or any misfolded protein, or lysosomal pathway for membrane or extracellular proteins. And because this is based on transcription and protein turnover, this time scale is hours to days. Next, short-term regulation. It modifies enzyme activity without changing the number of enzymes. Um, and first, it happens through three ways. First, substrate concentration. Reaction velocity increases at substrate level rise and up to the enzyme maximum velocity. So for example, glucokinase activity in the liver depends on how much glucose is around. Next, product inhibition. So here the direct product of the reaction inhibits the enzyme. And for example, glucose 6-phosphate inhibits hexokinase. And make sure to know that this is different from feedback inhibition, which involves a product from much later in the pathway. Next one, allosteric regulation is another big one. The effector binds at the site other than active site, and it changes enzyme shape and activity. This allows fine tuning of metabolism in real time. Next. And there's reversible covalent modification. So enzymes can be activated or inactivated by covalent modification, most commonly phosphorylation and dephosphorylation. So note that kinase add phosphate groups and phosphatase remove it. And the key amino acids here are serine, threonine, and tyrosine. And there's one classic high yield fact. Insulin always dephosphorylate key metabolism enzymes, which generally activates them. Next, zymogen activation. So some enzymes are made as inactive precursors called zymogen, and they're activated by proteolytic cleavage in order for the enzyme to get function. So a classic example is pancreatic protease. So trypsinogen is activated to trypsin by enteropeptidase. So clinical relevant example is like inappropriate activation of trypsinogen within the pancreas can lead to acute pancreatitis. Next, compartmentalization. It prevents conflicting pathway from interfering with each other. So, for example, fatty acid oxidation happens in mitochondria. Meanwhile, fatty acid synthesis happen in the cytoplasm. So this spatial separation is really important for efficient metabolic reaction. Do you guys have any question about lecture 6? Okay, I'll move on to lecture 7, which is about enzyme and enzyme kinetics. Next slide, please. So let's talk about what are enzymes. Enzymes are biological catalysts. They speed up uh, chemical reactions without being consumed. With, with the exception of ribozymes, they're all made of proteins. And high yield fact here is that enzyme never changes delta G, H, or final equilibrium. They simply lower the activation energy, allowing the reaction to proceed faster. And importantly, they work in both directions and they are reusable. And some enzymes require helpers. Inorganic helpers are cofactors, like iron for cytochromes or zinc for carbonic anhydrase. Organic helpers are coenzymes. So co-substrate like NAD plus that comes and go, or prostatic group like FAD that are permanently attached. So remember, co-substrate are like they're like Uber rides, and prostatic groups are like wedding rings. They stick around. Next, the Calus-Menten kinetics. Make sure to know the definition of Km, Vmax, Kcat. I'll briefly go over it right now. So Km is the substrate concentration at which the enzyme works at half its maximum speed. A high Km means low affinity. Vmax is the fastest the enzyme can go, depending on how much enzyme is present. And Kcat tells us how many substrate molecule each enzyme can convert per second. So think of it like enzyme's personal speeding rate. And substrate concentration can affect this kinetics, right? And how? Like when the substrate is much lower than Km, the reaction is first order kinetics. It speeds up proportionally with the substrate concentration. And the great example for this is GLUT2. It has a high Km and works best at the high glucose level and acting as a sensor in the liver and pancreas. And when the substrate is way above Km, the enzyme is saturated. So it's zero kinetic order. Example for this is GLUT1 in the brain, which ensures constant glucose uptake even when level fluctuates. Next, other factors that's affecting the activity. So the velocity increases with the enzyme concentration and with temperature up to about 37 degrees Celsius for human enzyme. And each enzyme has their own pH, like sweet spot. So pepsin works in the stomach at pH 1 to 2, while alkaline phosphatase prefers pH 9 to 10. So you guys should remember these numbers. Next. Uh, okay, now we're going to talk about inhibitors. First one is competitive inhibitor. So the competitive inhibitor looks like structurally similar to enzyme natural substance. So it competes for the active site. If enough substrate is present, it will eventually outcompete the inhibitor for the active site. So therefore, Km increases because you need more substrate to reach half of Vmax. And Vmax has no change because if you add enough substrate, you can still reach the enzyme full catalytic rate. And example drugs are statin and methotrexate. I think they're gonna test the drug name too, so make sure to memorize that. Next one, non-competitive inhibitor. So this inhibitor binds not at the active site, but at the allosteric site, changing the enzyme shape and making the active site less functional or inactive. So binding can occur whether or not the substrate is already bound. Substrate cannot outcompete this type of inhibitor. Therefore, Km has no change, but Vmax decreases. Yeah, and the next slide. So make sure to know like what it looks like on the graphs too. You guys can take a look at it if you have any question. Should be your email or yeah. Next. Did you skip one by any chance? Wait. Oh, can you go up like two?
One more.
Yeah, one more.
I have a question.
Yeah, sure.
Um,
So mine was for the inhibitor slides.
Yeah, I think two up.
After this, it's just the next lecture.
No, go back.
Um, Here.
Yeah, yeah,
Okay.
So like, what is the difference between irreversible and suicide since they both are permanent and bind
yeah.
covalently? I really don't understand the difference between the two.
Right. I skipped that part, so let me explain. So irreversible and suicide inhibitors. Irreversible inhibitors covalently bind and disable the enzyme permanently. Examples are cyanide and aspirin. And suicide inhibitors are very special. They bind the active site and they lock the enzyme out. Um, Suicide inhibitor produce the same kinetic effect as the non-competitive inhibitor, but the difference is that suicide inhibitor directly like binds to the active site. And your question was, what's the difference between irreversible and suicide?
Yes, is like irreversible only for allosteric sites, or what is the difference?
Um, I can take a look at it later. I actually don't know what's the difference between these two. They usually compare suicide inhibitor to non-competitive inhibitors, though. Does any of the TA knows what's the difference?
I think,
Uh,
oh, sorry.
yeah, go ahead.
I think suicide inhibitors bind to the active site whereas irreversible doesn't. I think it binds elsewhere. But
Maybe.
um, it cannot be reversed. I think it's just the difference in binding sites. Like one is to the active site itself and the other is elsewhere.
Yeah, maybe.
And I think there's also a question in the chat, Yuri.
What happened to KM and Vmax in irreversible? I don't think the lecture went over what happens. So they're not gonna test it, and then you don't need to know, I believe. Okay. Let me explain the allosteric effectors. KP, can you go down one more? Yep. So allosteric effectors, these enzymes have like multiple subunits and binding sites. K-type effectors change the enzyme affinity for its substrate by stabilizing the enzyme, while V-type effector changes catalytic turnover. So this graph A is V-type and B is K-type. And if they're positive effector, then it boosts the activity. If it's negative, then it suppresses it. These enzymes shows cooperativity and do not follow the Michaelis-Menten kinetics. That's the last slide for lecture 7. Next one, lecture 8 by Yvonne.
Hello. Hi, can you hear me?
Yes.
Okay. So we are going to start on this. Give me just a second. I'll pull this to the side. Okay, so we can go ahead to the next slide. The main part of this lecture, she just begins to talk about now we've moved on from the TCA cycle and we're now talking about the electron transport chain. So this portion, she starts off basically wanting to make sure that you understand the foundation for it, basically knowing that oxidation is an exergonic process. So energy gets stored in the form of ATP. There's a lot of information on this slide. Not all of it is really relevant because it's more so just like showing you the breakdown of how you get metabolized, how it goes from catabolism, you have your macronutrients becoming micro, how those break down, you get NADH, and then that gets shunted to create ATP. So that's kind of the summary of this whole slide. So we can go to the next one. So the next part basically just explains how electrons are transferred from the donor to acceptor. So these are just some rules and facts, a little bit of math. To understand like kind of like the foundation for it. So electrons get transferred whenever she talks about like reducing equivalents get transferred. She's talking about the electrons getting transferred. So they get transferred from the donor to the acceptor. Of course, the donor gets there's the donor is the reductant, acceptor is the oxidant, just terms to know. And then the main emphasis on this is that you should know that the redox potential, which is the E naught hash of the acceptor, will always be higher than that of the donor. So for example, it can be like the donor is -5, like -0.5, and then the acceptor will be higher than that, right? So it'll be more positive. So acceptor potential will always be higher. And that way the electrons can move forward. So if you have in the electron transport chain, it's going to be arranged from the lowest redox potential to the highest redox potential at the very end. So each time the electrons are being passed from like complex 1 to 2 to 3 to 4, the redox potential of the next complex in the lineup will always be higher than the previous one, so that they're able to accept the electrons. So again, just some facts here of the low electron potential being low affinity, high electron potential being high affinity. So the more positive you get, the higher it is. And so based on that, in the electron transport chain, the highest redox potential in the pathway would be oxygen because it is the final acceptor, and therefore it would have the highest E naught hash. Okay, so next slide. This portion just going over the electron transport chain. So sometimes it helps to have, I'm sure you guys have done this at some point in undergrad, but it does help to have a visual of the whole thing or like a video just to see how the electrons are being passed. So it's just electron transport starts when NADH, which is the carrier of the electrons, it'll bring its 2 electrons to the first carrier in the complex, which is complex 1. And then from there, you have 1, 3, and 4, which are going to be the proton pumpers. They're pumping protons into the intermembrane space. And then complex 2, which is on the matrix side of the mitochondria, it doesn't directly pump the protons, but it will help with complexes 3 and 4. So this is more important to just kind of know its function. You should know where the complexes are. You should know that 1, 3, and 4 are transmembrane. You should know that 2 is on the matrix side. CoQ is on the intermembrane side, is more superficial. So the schematic for that, and then, yep, so CoQ, which is the middle one right there, it's going to receive the electrons from both complex 1 and 2, and then it'll donate the electrons to complex 3 to then continue down the pathway. This becomes important later on when we're talking about NADH and FADH2 and why they're different. And eventually it gets pumped, all the protons get pumped into the intermembrane space through this process. And normally this inner membrane of the mitochondria is not permeable. So this is how we keep that intermembrane space really high proton concentration, and then we can shunt it all back in for ATP synthase to create ATP. Okay, next slide. This one is just a really quick same diagram, except it has the names listed underneath. For the purposes of the exam, you should know the names of all the complexes, like succinate dehydrogenase is complex 2. You should know that cytochrome C oxidase is 4. So they might be referred to as like different as their full names instead of just complex 1, complex 5. So you can ignore all the other stuff. Just make sure that you know the names of these things. And next. So I included a couple slides in here that we can just kind of skip through, but it's like you can look back on practice just to make sure that you know what the donors and acceptors, so where is each complex getting its electrons from, where is it giving it to? That's something that you should know for sure, just to make sure you have like the, the flow of everything correct. So we can just skip through the next couple one, and yep, and then we have term related to its function. Again, just practice just to make sure you know what each function is. And answers are next. And then for the ETC, here we have a full schematic of the ETC. This one is still the same thing, except it's written out with more descriptive terms. For this one, you do need to know like complex 1 FMN. Complex 2 FAD. You do need to know, complex 3 is the cytochrome B to C1, and then that complex 4 is cytochrome A, C. Why? I'm not sure. They will just be referred to as those names. So again, it's just the naming thing. And then the main thing that you want to get from this is just 2 things really. One, that copper is in complex 4 is something that requires copper. So if you have a deficiency in copper, know that that's something that will affect complex 4. And what else she had put here, which is the P to O ratio, it's basically just how many molecules of ATP get produced for each molecule of oxygen that you consume. So there's a difference between if you're starting with FADH2, which starts at complex 2, goes to 3, goes to 4. So if you start there and you see you only get 2 ATP for each 1 molecule of oxygen that you get consumed. So that's the 2-to-1 ratio. If you have NADH, you start at complex 1, and then you go 1, 3, and 4. And so you get 3 molecules of ATP for every 1 oxygen consumed. So that's the 3-to-1 ratio for NADH. So just kind of a fact that could come up in like a more simple type of question. And yep, so just make sure you know the directions of everything, where everything is going, where everything's coming from, and I don't know all the names here. Okay, next. This is a question, but we don't have to do it now because it's not really too high yield. It's not really high yield for this test because superoxides are important. They're just not important right now. I mean, ROS, reactive oxygen species, they're important. They're just not as important now, which is kind of something that was in the lecture. So you should know that in the electron transport chain, superoxide is produced. So we can go to the next slide. And again, all the information I have is in the speaker notes in case you want to review it. So superoxide is made in the ETC. It leaks out from complex 1 through FMN. It also leaks out from complex 3 from ubiquinone, just kind of more of a memory thing for this lecture. And then inhibitors of the respiratory chain. So for the test for this part, you should just definitely know anything that is highlighted in pink. So like in number 1, A and C, you should know like in number 2, B. You should know the names of all these drugs and then also which complex they're inhibiting, and also based on that, like basically where is this complex located? What is it getting stuff from? Where is it sending stuff to? And then yeah, so you should know the names of all the ones that are highlighted here. And then the next slide is a more laid out version of everything, where we basically see all the inhibitors versus the uncoupler, like the inhibitors of the electron transfer chain versus the uncouplers and kind of the difference between the two. So the main thing you should for sure know, any of the names that are listed here, you should for sure know them, the drugs, and then what complex they're related to. For the inhibitors, the main function is that because they're stopping something in the path of electron transport chain, so they're blocking electrons moving through the chain. So it could be blocking at complex 1, 2, 3, or 4. I don't know why I put 5. Oh, yeah. Okay. And then, so because they're blocking electrons from moving through the chain, it essentially reduces the function of the electron transport chain. And so because it's not moving forward, you're not using oxygen because oxygen is the final acceptor, right? So if you aren't, the electron transport chain isn't moving, the oxygen's not going to get used. So your ATP synthesis will also decrease. Versus uncouplers, which will increase the permeability of the inner mitochondrial membrane to protons, which we mentioned before. Normally it's not permeable, but if you increase the permeability, all that pumping that we're doing is going to be for nothing. So for this, you need to know that because we're losing that gradient, and all the protons just keep leaking out. In this process, the electron transport chain keeps going, which is why there's an increase in ETC function. So it keeps going through 1, 2, 3, and 4, keeps shunting the oxygen, and then it keeps trying to do like the proton pumping. But so it keeps running and you keep using O2 because eventually the electrons get to oxygen and then it accepts it. So your O2 consumption is high. And the issue is because now we've increased the permeability, we don't get that proton gradient, we can't shunt it through ATP synthase. And so you just accumulated all this energy, and all this energy has nowhere to go. So it ends up getting released as heat, and then your body temperature increases. So the main importance of this slide, know all the drugs that are listed on here, just know this slide in general, and then relate it back to the function in terms of inhibitor, ETC function decreasing, oxygen consumption decreasing, ATP synthesis decreasing. Uncoupler is essentially the opposite, but you're still having that decreased ATP synthesis. So very important slide. Last thing is going to be the last slide. So, I guess not here. Okay. There is a slide that's weird that is in here, but I guess you can't see it. Okay, I can see it on my end, but there's two more things that she talks about in like the lecture, which is just the carbon monoxide poisoning and the cyanide poisoning. For both of those, it's mainly just about knowing the facts that come with it. So for example, carbon monoxide poisoning, you should know kind of what they would provide to you in the stem of the question. Like camping, they're in a closed tent, they have a lantern burning. You would think carbon monoxide poisoning, and then you would kind of have to know the facts that are associated with it. So carbon monoxide binds to the iron in complex 4. You should know that a symptom is cherry red cyanosis, and you should know that you treat it with 100% oxygen so that you can push out and replace the carbon monoxide with oxygen instead, because when carbon monoxide binds to the heme molecule, it doesn't let the oxygen offload, which just means that when carbon monoxide binds heme, the other oxygens stay on and then they cannot be used. So that's kind of why you end up with the cherry red cyanosis. So all the facts, once you see the slide, it's the same thing as what's in the lecture. The blue text is very important. It's just kind of facts that you should know if she doesn't really like you don't have to go too in depth into this. It's just more so like, you should know which direction the oxygen binding curve shifts. You should know that it goes from sigmoidal to hyperbola, just stuff like that, just general things like that. And then same thing for the cyanide poisoning. You're just wanting to know kind of what is it that they would include in the stem of the question, cassava-based meal, sodium nitroprusside and medication. So that's something that they would give to you. You should know this. Cyanide binds again in the heme of cytochrome A3, prevents O2 rejection, and then what you basically treat it with, which is methemoglobin. So you want to create methemoglobin. So it's just very simple stuff. Just know like what they would present to you in the question, and then know kind of a couple of facts that they list on the slide, and then just know the treatment. So that's basically a summary for those two. And I think I'm done.
Yeah, I'm sorry. I'm not sure why it's not showing up on this thing, but it should be on the presentation, and I think you all have access to it. So on your own time, whenever you have the chance, feel free to go over those.
Hey guys, uh, hey KP, could you just go to the 2 slides ahead? That'll be my first one. Perfect. So with this one, there's not really much for me to explain. It's just you're simply gonna have to memorize it, but I can tell you some of the high yield stuff. So for example, I remember pretty sure we got tested on it, or it's like a high yield concept at the GPCR. It's a transmembrane structure with 7 alpha helical domains. So I think that's important to know. And just literally any of these plasma membrane receptors, it's important to know. So for example, like ones that cause phosphorylation, tyrosine, serine, and threonine, those are important to know. And I think even last year they tested for the intracellular receptors, like for example, cytosolic, what would be a ligand for it? So something that's cytosolic, it's going to be lipophilic. So like a vitamin D or like a steroid hormone would bind to that. So I'd say that's important to know. And insulin uses a receptor tyrosine kinase. That's also important to know. And yeah, we can go to the next slide. So for this one, once again, you're just gonna have to memorize it. All this stuff is really high yield, important information that we still get tested on even to this day. So just literally know this whole slide. And I also included a mnemonic. It's called KISS Quick, and it tells you what each alpha 1, alpha 2, beta 1, beta 2 do. So the Q is for alpha 1, and that's going to do the GQ IP3 cascade. The I is for alpha 2, which is in VGI. It's going to inhibit that, or it's going to inhibit the GTPase and decrease CAMP. And then the GS for beta 1 and beta 2 are both going to increase CAMP. And I don't think you really have to know the quick part for this class, but definitely for physiology, you have to know it. So you might as well just learn it right now. And yeah, so just memorize this. I think it's a really important thing to know for the exam. Yep, KP agrees with me. All right, yeah, next slide. So once again, not a lot to explain. You're just gonna have to memorize these steps, but I just included something that I can almost guarantee will be on your test. It's going to be sildenafil or Viagra. So how this works, it blocks phosphodiesterase 5, and it's going to increase your CGMP. So that will probably be the answer on your test. That increases CGMP. So I'll definitely know that. So and Viagra is obviously used in erectile dysfunction, so it's going to vasodilate. And like I said, increase CGMP, which is the most important thing. Go on to the next one. And I just included another clinical correlate of the GPCR signaling. So there's chlorotoxin and pertussis toxin. So with both of these, there's going to be an increase in CAMP, but how it gets there is different. So in pertussis toxin, the GI subunit, which is the I in the kick mnemonic, it's the GI is going to get inhibited, so the CAMP is going to keep on getting produced, and that's how it's increased. Chlorotoxin is different because the GS gets ADP ribosylated, and GTPase is constantly bound, so you're going to keep on increasing CAMP. So one of them inhibits the inhibitor, and the other one just activates part of the cascade, the GS. And once again, I can almost guarantee that one of these will definitely be on your exam. Yeah, next slide, thanks. And this is also kind of similar to the GPCR. Just another thing you're gonna have to know. I heard Yuri mention how insulin dephosphorylates a key enzyme, so that's like another concept here that can be applied. But yeah, I would also know which receptor is going to be where. So like, for example, epinephrine can be used in every single one, alpha 1, alpha 2, beta 1, beta 2. And how they get degraded. So they'd get degraded by phosphodiesterase here. And like I said in the slide before, Viagra is going to inhibit the phosphodiesterase to keep elevated CGMP levels. You can go to the next slide. Thanks. So once again, you're going to have to know another cascade. So with this one, the unique thing is that the receptor is going to already be dimerized. So before insulin binds, the receptor is already dimerized compared to like when a growth factor binds, it's going to be monomeric and then it dimerizes. But this one is pre-dimerized in the RAS-MAP insulin pathway. Another important thing I remember that they tested was, so for example, when you're exercising, why it's so beneficial is because the GLUT4 receptors get transported to the skeletal muscle and glucose is able to be taken up. And I remember they asked a question on that. So I wanted to include that. And yeah, insulin also dephosphorylates the key enzyme, which I already said. Next slide, please. So these are just some more clinical correlates. So I included, for example, when RAS gets mutated and it's constantly activated, as seen in the bottom right here, it's going to cause neurofibromatosis. That will also probably be a test question. So I'd say know that as well as the other 3 clinical correlates. And that's all I got. Thank you.
Great. Well, that's been a lot of information for the past few slides, and I will wrap things up with the last 2 lectures, lectures 10 and 11. These 2 lectures are a bit more light in the fact that it's more stuff that you probably already know about diet and like different facts about carbohydrates, proteins, and fats. The last few lectures with Alex, Ivan, and Yuri have a lot more detailed information, a lot more information in detail that you should definitely pay attention to. So I'll just quickly wrap up the lecture and hopefully we'll have some time for any questions you guys have. So lecture 10 is about different macronutrients. It's a pretty word-heavy lecture. I don't know if you guys have taken a look at the lecture slides, but I've kind of summarized it and put down some key points, some things to focus on. Definitely know BMR, basal metabolic rate, and how it's measured at different parts of the day. Know what is affected by it, so like body temperature, age, gender. Definitely know what affects the BMR. Know the different clinical correlates that are associated with BMR. So like hyperthyroidism, hypothyroidism, different varying levels of TSH, thyroid stimulating hormone. Um, know like how that affects BMR. One increases it, one decreases it. And here are some clinical correlates I included. Graves is an example of hyperthyroidism. So that usually leads to increase in BMR because there's an increase in thyroid stimulating hormone. Um, and usually it helps you burn more fat. Whereas Hashimoto thyroiditis is an example of hypothyroidism that usually lowers your BMR and helps you or makes it so you don't burn as much fat as well. Moving on, no thermic effect on food is usually pretty negligible. Physical activity is the biggest component of measuring BMR, and you usually multiply that with the BMR or the activity factor to find the total energy expenditure. And that's a pretty much all you need to know regarding like energy expenditure and BMR for the first part of the lecture for lecture 10. And then we move on to different macronutrients like carbohydrates. Definitely know the difference between like monosaccharides, disaccharides, polysaccharides. Know the differences between glucose, fructose, galactose, sucrose, know which ones are monosaccharides, which ones are disaccharides, and know like the constituents of disaccharides. Um, know how to find the glycemic index. This is pretty small. I don't remember getting tested on this too much on exams last year, but just something to keep on the back of your mind. Know the difference between soluble and insoluble fiber. Soluble helps you feel full for longer, while insoluble fiber helps with stool movements. Um, and then I included some information on the recommended dietary allowance for protein. So see what happens when you take too much protein or when you don't have enough protein. And I included some clinical correlates that you'll see with deficiencies in malnutrition and in protein deficiencies, kwashiorkor and marasmus. These are two very important clinical correlates that you will get tested on. So make sure when you're looking through the lecture or watching the lecture videos, you keep an eye out for these two conditions and try to learn as much as you can about them. They will pop up on your exam. Um, and for the last part of this lecture, know the different types of diets. So like a Mediterranean diet versus like a ketogenic diet or a paleo diet. Know the different types of fats, saturated versus trans fats versus unsaturated fats. Um, and know the difference between omega-6 and omega-3. And when you look over um, lecture 10 like over through lecture videos or lecture notes, you can kind of use this slide as a guideline of things to focus on and what I thought were the highest yield points of the lecture. And most of these will probably show up as questions on your exam. I'm going to move on to lecture 11 really quick. So with this one, going through the lecture was if I don't know if you guys are used to like the way that different lectures are written by different professors. The last one is written by Dr. Papa Nicolau, I think. And his lectures are usually pretty word heavy, whereas this one is done by Dr. Binstock, um, who has a lot of assessment questions throughout and also like very simple slides, I guess, just to what I mean is like there's like very few words per slide, and so it's pretty easily digestible. So in addition to um, so going off of that lecture here, some questions that I made, and when you guys go through the lecture yourselves, through the lecture videos or through the slides, here are some questions that I prepared that some like first order and second order questions that I prepared that I feel like once you're able to answer these, you should have a pretty good understanding of the lecture, and um, you should it should answer most questions on the exam. So I'm just gonna quickly go through them. The main source of carbohydrates in the world, I think you'll see is usually like rice, or pasta, some kind of grain, and that usually has a lot of glucose in it. You'll see it's labeled in red. As I mentioned in the beginning of this lecture, a lot of colored notes, and in lecture slides that you saw with the other TAs, all the colored information in blue, red, pink, green, those are all very important, very high yield. Um, know the organs used in carbohydrate digestion. So like the mouth, saliva, stomach, and all that. Um, and here's a second order question that I included, something special about protein and saliva. So um, if you guys aren't caught up, I'll just go over it very briefly. So saliva has salivary amylase that begins digestion of food from polysaccharides into monosaccharides. The special thing about salivary amylase is that it can't digest disaccharides. So like small little details like that are also something that you want to keep in mind when you look through these lecture notes. Um, I had then you move on to questions about SGLT and GLUT transporters. I had a couple of questions about those. Make sure you're able to understand those. They're very important. The different SGLT1, SGLT2, know where they are and what they're used for. The different GLUT transporters, there are five main ones that you want to know: GLUTs 1, 2, 3, 4, and 5. Know the different tissues that they transport to, and I think Yuri or Yvonne touched on it in their lectures or in their slides. Like GLUT1 transports uh, uh, glucose to the brain. Um, GLUT2 goes to um, skeletal tissue, I believe, and so on. And know the different digestive deficiencies. So what happens when you have issues digesting glucose, or like celiac disease, or what happens when you have issues digesting like fats or proteins. So all those clinical correlates are fair game for the exam, and they probably will be tested. And with that, that concludes the lecture for today. Um, if you guys have any questions, I'm ready to take them. All right. Well, uh, you guys have access to the presentation, and